Hello, everybody. You have successfully made it through one unit, Unit 8, and now we're bringing it back all the way to the beginning to Unit 1, where we are going to talk about the chemistry of life. So we've taken a look at the big picture, to ecosystems, to all of the interactions that exist between living and non-living pieces of our biosphere. And we are going to zoom in now. We're going to take a look at all of the molecules and elements that make up these pieces and allow them to function in the grander scheme. So in this video, we're going to review some of the major chemistry items that you will want to put into your tool belt to better understand a lot of the more complex goings on that we'll hit in future units. And we are going to spend a lot of this video focusing on water. Water being one of the key factors that allowed life to rise on Earth and continue to survive here today. So we will get started today with some essentially real quick chemistry review. So we know that anything that has mass and takes up space is composed of matter, and that matter is made up of elements. So the elements that we are going to be looking at in this class are just a few out of all of those available on the periodic table, because these are the elements that are involved in all of our biological molecules, our major structures that make us living organisms. So we are going to focus on these key elements: oxygen, carbon, hydrogen, and nitrogen. And aside from just looking at them in their individual elemental atomic state, we are going to consider what happens when we combine them together, when we create compounds. So you hopefully recall from previous classes that compounds are fixed ratios of two or more elements that are bonded together, and we can form all sorts of different structures, from very simple diatomic structures to more complex geometries. So we're going to see things like salt, water especially, simple sugars like glucose, fructose, oxygen gas, all of these things that play relatively large roles in our survival on Earth. An important thing to note is that when we take these individual elements and we combine them together to form compounds, they take on what we term emergent properties. So they have characteristics that didn't exist when they were individual elements. They have new functions. And so these emergent properties are what we're going to see play a large role in these compounds being the basis for life. So let's talk a little bit about elements, starting us off. Those four elements that I mentioned, those make up 96% of all life on Earth. These are going to be ones that you will want to remember, especially when we go into the next video talking about biological molecules. Now, these all fall into the category of essential elements, ones that we require in large amounts in order to survive. The essential structure of our biological molecules are composed of these four elements, but we do have other elements that play other roles in terms of our homeostasis and our ability to survive and withstand environmental change, and those are considered trace elements. So those are going to be things that create concentration gradients, is one of the examples that we'll talk about, that allow our neurons to communicate and send signals. So trace elements are also very important. We just require them in smaller quantities than our essential elements. They're all needed to survive. It's just how much of these elements we need really makes the difference between whether we consider it to be essential or trace. So bringing it back to the structure of these elements, you should hopefully recall that the atom is the smallest unit of matter that still retains all those lovely properties of our elements, and that atoms themselves are made up of subatomic particles. If you're just now taking a chemistry class, this may be something that you've already gotten into. It may not, but either way, this is a good review or introduction to the structure that we see at an atomic level. Now, our atoms are composed of two essential regions. We have the center of our atom, that is the atomic nucleus. So nucleus means, usually refers to a kernel. So it is the central structure in our atom. And inside the central structure, we find our protons, which are positively charged, and our neutrons, which are neutrally charged. And these are held together by a nuclear force. So they really pack in there, and they don't like to break down, although we're going to talk a little bit about radioactivity here. And outside of our atomic nucleus, floating around in orbital shells, which whether we look at a Bohr model like I have here, or a quantum model where we have more of those clouds, we just want to focus on the idea that the electrons are not bound into that central location. These are our negatively charged subatomic particles, and they are our big players when it comes to being able to create compounds. When we take a look at atoms, we can define some of the characteristics that these atoms have based on the number of subatomic particles and the mass of those individual particles. So you'll hear a couple of different terms that we're going to throw around, things like atomic number, which tells us how many protons we have in a nucleus. If we have a neutral atom, the number of protons is also the number of electrons. And then we can consider things like the mass number, which is the number of protons plus the number of neutrons. These are the two subatomic particles with, mass that we actually consider to be mass. Electrons are negligible, so we really don't include them in this piece. And so the mass number is going to be the sum of our protons and our neutrons, essentially the mass of the nucleus. And when we look at something like the atomic mass, we're taking the true mass. So we're considering all of the different isotopes that exist of that particular element. So all atoms of a specific element have the same atomic number. They have the same number of protons. Otherwise, you have a different element. But they can have more or less neutrons depending on their structure. And so that's why the atomic mass usually differs slightly from the mass number of an individual atom, because we are considering all the different versions of that mass that exist on Earth. As mentioned in the previous slide, we want to consider isotopes. Isotopes, which iso means equal, tope meaning form, like topic. These are forms that have an equal number of protons, but may differ in the number of neutrons. And even though the masses themselves differ, they're going to react the same in chemical reactions. And we as biologists love to use isotopes in a couple of different ways. Some of these may involve radioactivity, which if you add too many neutrons to an element, neutrons are like this stabilizing force within the nucleus. If we add too many, we start to see that nucleus isn't necessarily the most structurally sound anymore. So we may start to release particles and energy, some sort of nuclear decay. And so we take this radioactivity that we get in some of these isotopes and we use it to our benefit. We have three major uses that I'm going to highlight here. We use isotopes to perform what we call radiometric dating. So radiometric dating allows us to determine the absolute age of a specimen by looking at the amount of carbon-14 that it has. We can use it in medical imaging, so we can inject radioactive substances into all the different parts that we want to look at, and when we look at it using some specialized technology, we can see changes at that level that we would not be able to see without the radioactivity present. And we can trace different elements through metabolic processes. I've highlighted here one of the experiments that they used to help identify how DNA replicated, and that was by using a heavy and a light nitrogen. So we have our different types of nitrogen that we can follow through the process and really look at how is this process taking place. Essentially, these are tools that we can use to zoom in on things that we would otherwise not be able to see. And I personally think that that is really cool, and hopefully you do too. I mentioned when I was going very quickly over the basic structure of the atom that electrons are really what does the majority of the work. Because they're not bound to the nucleus and they're able to move, they are what are directly involved in all of the chemical reactions that we are going to look at over the course of this year. And one of the important things that gives them this ability is because existing in those different shells or those different clouds, they have a certain amount of potential energy. Think like you have a ball on the top step of a stairwell. It has a certain amount of potential energy. That energy could be converted into kinetic energy if it were to drop down to the next level. It would have less potential energy on the next stair step. We see something very similar when we look at electrons in terms of their location outside of the nucleus. They have less potential energy the closer they are to the nucleus because they are attracted to those positive charges that we have in the protons. And so the farther away they are, the more potential they have to return to a closer spot, essentially. So when our electrons want to move between different levels, they're going to have to either absorb or lose energy equal to the difference in that potential energy. I do want to emphasize that even though I'm kind of giving this crash course on chemistry, this type of information is really just background. We should not have any questions about electrons nor their potential energy or how they move between shells, but just as a piece to help you understand why it's so important when it comes to chemical reactions. Because really, even electrons, some of them are more important than others. And when we are the farthest from the nuclear shell, when we are in that valence shell, that is where we're going to have all of our activity. So we term valence electrons as the electrons that are in our outermost shell, and these are the ones that are going to really interact in those chemical reactions. And the reactivity of our element largely depends on the number of valence electrons that we have. So you will hear a term called octet rule. And the octet rule is all about how many electrons we have in that valence shell. Electrons love to have a full valence shell. And after we move past the first period, our valence shell can hold up to 8 electrons in that first level. There are more beyond this. It can get more complicated, but for now, we don't need to focus On the more complex pieces, I will leave that for chemistry. But it is important that we want to complete that valence shell, so we want to get to 8 in any way possible, or 2 in the case of hydrogen. So as mentioned, it's the electrons that are really doing all of the heavy lifting in the formation of chemical bonds in order for us to make these compounds. Now, the reason that the electrons want to do this is because the atoms seek stability, and they get stability from having a complete valence shell. So some of the ways that atoms can complete these valence shells are by forming chemical bonds. Now, hopefully, none of these chemical bonds that I'm presenting on this slide are unfamiliar to you. And hopefully you guys remember from chemistry, or if you haven't had chemistry yet, or you haven't got there yet, just know that there are all sorts of properties across the periodic table that atoms have based on their structure. And one of these is termed electronegativity. Now, electronegativity is the amount of pull that the electrons have towards the nucleus, so towards all of those protons that they are positive, opposites attract, all that. So the type of bond that's going to be formed when our electrons interact with each other determines on the electronegative difference between the two atoms involved. So we'll essentially find the difference, we'll subtract one from the other, between these two atoms, and that will give us an idea of where our atoms run along this continuum. So the smallest amount of electronegative difference, if our atoms are really close together in terms of their electronegativity, these will form what we call nonpolar covalent bonds. Essentially, our atoms will share the electrons equally. If there's a slight difference between the electronegativities of our two atoms, somewhere between 0.4 and 1.2, we will get what's termed a polar covalent. So one of those atoms is going to be a little bit of an electron hog. And what that is going to create is what we call a dipole. A dipole is a representation of partial charges between our atoms. And then if we have a severe difference between the electronegativities of our atoms, we will get complete differences in charge. This is where we lose electrons, we gain electrons, there's no sharing involved. Everyone is very greedy or just really passive about where their electrons are. And so these are fully charged ions, either cations if they're positive or anions if they're negative. We're going to detail some of these different chemical bonds here on a few of the next slides, but the reason that these different kinds of bonds exist is all due to this electronegative difference. So this is just going to be a little bit of a recap across all of these to what I mentioned on the previous slide, but hopefully we recall that in general, a covalent bond is produced by two atoms that are sharing a pair of valence electrons. They want to create that perfect octet within their valence shell, and so they kind of have shared custody of these electrons in order to feel stable and secure. When we create a compound using covalent bonds, we term it as a molecule. So these terms are not interchangeable. It's kind of one of those square rectangle situations where all molecules are compounds, but not all compounds are molecules. Now, when we take covalent bonds, we can split them into two categories based on those electronegative differences. Here with our diatomic molecule of oxygen gas, O2, we have a nonpolar covalent bond. Obviously, both of the atoms of oxygen have the same electronegativity. So their electronegative difference is 0. Therefore, their electrons are shared equally. They are immediately between both of the atoms so that both atoms feel like they have that full grasp on those electrons. Now let's say that we don't have a perfect zero electronegative difference. Well, then our covalent bond becomes more of a polar covalent bond. Polar covalent bonds have slight, I use the term significant here in the description, but really significant is more ionic. So these are slight differences that borderline on significant, and therefore the electrons feel more of a tug towards one of the atoms than the other. And so we're sharing a little bit unequally, but we're still sharing. So this creates these partial charges, creating the dipole. So some atoms have a partial positive charge, whereas some atoms have a partial negative charge. You can see here our example of a polar covalent bond. The example to end all examples is going to be our water molecule. The oxygen has a stronger pull on those electrons because it has a larger electronegativity. And so it has a slightly negative charge compared to our hydrogen atoms, which are a little bit more loose with their electron affinity. So they get a slightly positive charge. Now these partial charges are going to play a big role when we talk about the properties of water, so stay tuned for that. But we do have one last type of bond that we want to look at, those being our ionic bonds. So ionic bonds are formed when the differences between the electronegativities of our atoms are so great that essentially we can just throw electrons away for some cases and take electrons completely. No sharing involved. Now, ionic bonds are formed between a metal and a nonmetal. So here we have sodium as our metal, chlorine as our nonmetal, and when they either give away or take electrons from the universe, they don't have to swap with each other. I do want to make that clear. They will become what we call ions. So these are atoms with charge. Now you can either have a positive charge and be a cation. I always refer back to what my chemistry teacher used to help remember, is that cations are positive. Yes, I will show myself out for that dad joke. It's okay. And our anions are negative. Now, within more complex molecules, we have all sorts of different interactions between our atoms. And these interactions, which are our weak bonds, these are easily broken, but they are influential to the overall structure of these more complex molecules. These are bonds that help to reinforce the shape and connect different molecules together. And one of these is going to come up way more than the other one, and that being hydrogen bonds. Hydrogen bonds are formed between the partially positive hydrogen atom and another electronegative atom. It doesn't always have to be oxygen. Sometimes it's nitrogen. Sometimes we throw other elements in the mix, but usually oxygen and nitrogen are the ones that we want to look out for. Now, hydrogen bonds play an influential role in a lot of our larger, more complex structures. We'll come back to this when we talk about nucleic acids. And not to dismiss van der Waal interactions, but these are interactions that help molecules kind of stick together or create kinks and bonds within larger structures. They're going to come back when we talk about protein structure, specifically the tertiary structure of our polypeptides. But they don't get as much love as the hydrogen bonds. Now, with the weak bonds helping to reinforce these shapes of our large molecules. It should stand to reason that shape is an extremely important part of the function of a molecule. You guys are going to probably get really tired of any biology teacher kind of hammering this note in, but structure determines function. So the size and the shape of our molecule are key to how they function within a cell. And this is because the structure determines whether or not we're able to bond with particular receptors for some cases. In terms of we have complementary structures, so we kind of fit in there perfectly. And we disrupt this structure, well, we throw off the entire system. And so we'll talk about some of the different ways that we can disrupt things and we can cause them not to work a little bit later on in this unit. But it's also important to note here that because structure is such an important piece of our functional anatomy, we're able to create things that mimic structures in order to get very similar effects. So you have here an example of a natural endorphin, and we can compare its structure to a lab-produced morphine. So we get the same effect from both of these pieces because they're both able to fit into the same receptor. But one of these is produced naturally within our bodies, and the other can be introduced to the system in order to mimic those effects. Okay, the last little bit of chemistry that I'm going to hit you guys with so that I feel like we have a good foundation going into a lot of the next few units is that of chemical reactions. Remember in the previous unit, Unit 8, we talked about all of those nutrient cycles that are pulling different elements through the biosphere in all sorts of different ways, and these are managed at each level by chemical reactions, where we have these changes in the composition of our matter. So we can start with one piece and we can convert it to something else, something that might be more useful. Now, chemical reactions play a key role when we talk about the metabolism of an organism. Metabolism being the sum of all of the chemical reactions that are taking place within an organism. And we are going to highlight a couple of different chemical reactions as we go through this unit and some units to follow. So it's important for us to understand generally what a chemical reaction is. Well, a chemical reaction is a process where we take reactants, what we're putting into the chemical reaction, and we are converting them in some way using energy to create different compounds. Now, what's important to note here is that matter is conserved. We do not lose any atoms. We do not create any atoms. This comes back to the conservation of matter, so that nothing can be created or destroyed, only rearranged and redistributed. So everything that exists in your body right now on an atomic level came from something else. So whether that be from the food you ate or from the initial cells that came from your parents, we can trace it all the way back to the stars, but I won't pull too much physical science in here. Now, what's important is to note that reactions are limited, and they are limited mostly by the concentration of their reactants. We can only do so many reactions based on how much product we have to work with. So if we only have 4 Legos, we can only do the work of 4 Legos. And so if we increase the number of Legos, we can create more structures, Legos are going to be one of those things that I kind of come back to because they are a really good way to kind of visualize what we're working with here. If we increase the number of Legos, the more likely they are to come into contact with each other, and in a molecular level, they collide because they're constantly moving, and these collisions can cause reactions and the formation of products. So as we decrease our number of reactants within our substance, we reach essentially the limit of the creation of our products. One of the things that is going to be equally important is understanding that all chemical reactions, to some degree, are reversible, that we can take products and we can use those products to create the reactants, which, of course, when we're going in the opposite direction, we swap the name. So our products become reactants, our reactants become products, and we can go back and forth in this kind of dance between which direction, either forward or reverse, that our reaction is taking place until eventually we reach an equilibrium. Equilibrium means that both the forward and reverse reactions are occurring at the same rate. So essentially, it looks like we're standing still. We have a constant concentration of reactants and products because they're being created in equal rates at the same time. We'll talk a little bit more about dynamic equilibrium later on. But in this case, I just wanted to introduce you to the concept that we can move in both directions and that we can reach a constant creation of both our products and reactants at the same time. Now for the part that you have all been waiting for. Really, this is the start of Unit 1. Is the beginning discussions on water and its structure and all the lovely properties that emerge from this structure. All organisms on Earth require water in some form, whether they live in the water and use it in order to move or reproduce, or we contain water that allows all of our different reactions going on within our cells to function. So looking and understanding water's structure and the role that it plays is going to be very important. As mentioned within the chemistry review, our water molecule is formed from 2 hydrogen atoms and an oxygen atom, covalently bonded. But the electronegative difference between those 2 types of atoms is to a certain degree that we create a polar covalent bond between the atoms. This causes the formation of a dipole, which creates a partial positive charge on our hydrogen and a partial negative charge on our oxygen. And this will allow our oxygen and our hydrogen to interact with other water molecules to create hydrogen bonds. Now, these hydrogen bonds are the reason that all of our different emergent properties are so important and so influential in the grand scheme of things. So I will mention hydrogen bonds a couple of different times. I will mention electronegativity a couple of different times. Hopefully that conveys to you the importance of our understanding of these pieces going forward. So, emergent properties coming from the polar covalent nature of our water molecule and our ability to form hydrogen bonds from that structure. These are what we're going to focus on throughout the remainder of this video. These properties play a bunch of different roles in allowing life to exist on Earth. Very important. So we are going to highlight a couple of these. Cohesion, which also kind of brings us to the idea of surface tension, not to be confused with adhesion. And we will break down the difference in those scientific roots, which allows capillary action, also very important. And then we get into some of these properties dealing with heat, high specific heat, high heat of vaporization, which allows us to look at some evaporative cooling. We have a low solid density, very unique compared to other molecules, and the fact that water is a nearly universal solvent. So we will start with cohesion. So co is a root which means with or together. So cohesion refers to the attraction of water molecules to other water molecules. It's what causes water molecules to remain close together. And it contributes to our ability to pull water up a tube and work against gravity. So plants really appreciate this. Whenever you use a straw, you also really appreciate this. Whenever you observe a meniscus or a water droplet, we have these hydrogen bonds to thank for holding that water together, or from holding water to other structures. So cohesion is loosely related to adhesion, ad meaning across. So these are attractions between the water molecules to other polar or ionic molecules. So the ability for our partial charges to be attracted to the opposite partial or full charges in another molecule. This also allowing us to pull up these narrow passages and tubes because we're able to essentially climb up the walls by attaching little by little to other structures. So both of these come into play when we look at how plants are able to move molecules of water up from the roots to the highest points in their structure. There is also a little bit of osmosis at play, but we will save that for Unit 2. Don't worry about osmosis. So here we have kind of a zoomed in version of the interactions between adhesion and cohesion, where we're dealing with transpiration. So transpiration is the movement of our water through a tube, whether that be from xylem or phloem, and the xylem vessel has some attractive properties to it itself. So it has partial charges that allow the water to latch on, and water is also able to latch onto itself. So they're attracted to each other with those hydrogen bonds, and these create a force, a tension that pulls our water molecules against gravity up our structure. Very important for things to be able to survive when they don't exist in water. Instead, the water is inside of them and allows us to pull different materials through our systems and get all that benefit of multicellularity at the end of the day. Moving away from cohesion and not moving away, I spoke too soon. Here I have a diagram that helps to show surface tension. So surface tension is how hard it is to break the surface of a liquid. And we get these nice water droplets because of the attraction between our water molecules. So you can see in this zoomed in version model of our water droplet that our water molecules are able to form multiple attractions to other molecules. And so they are able to stick together essentially. And we have to create enough force to break these bonds, these hydrogen bonds between our water molecules in order for the surface to be broken. With that being said, now I get to move away from cohesion and adhesion. And we'll start with some of the energy that is going to be important. So you may recall if you've ever visited a coastal region, that the closer you get to a large body of water, the cooler it feels. And that is due to the fact that water has a high specific heat. Well, what does that mean? What is specific heat? Specific heat is the amount of heat energy that needs to be added to a system in order for 1 gram of that material to increase by 1 degree Celsius. So water has a very high specific heat, meaning that it can absorb a lot of heat energy before it starts to change its temperature overall. And the reason that this is because the heat absorbed by the water breaks those hydrogen bonds and is released when hydrogen bonds form, because bonds work in a little bit of a different way. So what happens is that as we absorb the heat, we can release the heat as cooler air and increase that cooling effect, which is similar to the evaporative cooling that we see in our own bodies. So this comes back to what we call a high heat of vaporization. So not only does it take a lot of energy for water to change its temperature, but it also takes a lot of energy for water to be converted to a gaseous state. So it allows water to cool surfaces as a liquid before it evaporates, allowing for evaporative cooling. Now, this heat helps to stabilize the temperature in our large bodies of water, our oceans. It also helps to allow us to cool down when temperatures outside of our body are too great. So if you've ever found yourself sweating because it's very hot outside, well, you have evaporative cooling happening in real time, allowing you to cool down before that water evaporates. So stepping away from heat and thermal energy, we're going to look at structure. So those hydrogen bonds that are formed between our different water molecules, those look a little bit different depending on the state our water is in. When we're in a liquid state, the hydrogen bonds hold water closely together, okay? And that closeness allows for all of those adhesion and cohesion that we previously mentioned. But when it starts to freeze, when it gets cooler, those hydrogen bonds expand. So they hold water molecules farther apart. So we create this crystalline structure that gives us a lower density. You may recall from previous science classes that density causes objects to sink or float. So one of the most important things that water is able to do in order for it to survive some of these big temperature changes is that when it reaches a solid state, when it freezes, the density decreases, allowing it to float. So that floating solid allows the warmer water underneath to maintain that nice consistency that allows organisms to survive and thus withstand those environmental changes. Another property, and this will be the last property that we'll really highlight here, is that water is a versatile solvent. So what is a solvent? A solvent is what you dissolve things into. And that is because due to the hydrogen bonds that it's able to form, it's able to break apart substances that have charge, that have those partial charges, either partial or full. And so we can take a solute, whatever we're looking to dissolve, and it will break apart within that water as a solvent and create a solution, a homogeneous mixture of that substance. So we're going to take a look at some of the different solutes that we'll deal with based on their particular polarities. The first being our ionic compounds. So when we have those perfect positive and negative separate charges, well, we introduce those crystalline compounds into our solvent, and water is able to break apart those positive and negative charges and form hydrogen bonds between the respective complementary parts of their structure. And so we get these pieces called hydration cells. So hydration shells are able to kind of wrap around our ionic bonds. This is what allows salt to break up so nicely and so evenly within water, because we're able to break apart that ionic structure and create these hydration shells, at least until we run out of water molecules, when we've reached our peak concentration. Now, it also helps to create opportunities for non-ionic polar molecules, as we can have some of those partial charges also interacting with water. So here we have a larger molecule, and while we're not able to break it apart as perfectly as we were the separate charges in our ionic bonds, it does help us to dissolve it in water, because we have smaller regions within that molecule that have properties, polar regions, partial charges, that give it the ability to adhere to our water molecule. So sugar being a good way, although we can increase the solubility of our sugar by increasing the temperature, because we have more opportunity for those molecules to interact with each other. So just coming back to those terms, hydrophilic and hydrophobic, which I mentioned, this refers to the ability of a molecule to either be attracted or repelled from water. And the important thing to remember here, one of the big pieces, is that like dissolves like. So opposite charges attract, but if we don't have any charge, we have nothing to be attracted to. So if we are hydrophilic, breaking down that word, hydro referring to water, philic coming from the Greek philia, or to love. So these are molecules that are attracted to water, either because they are partially or fully charged, so our polar and ionic molecules, polar molecules and ionic compounds, these are attracted to our water because of their charge. Hydrophobic, on the other hand, phobic coming from phobia, or a fear of, is where we are repelled by water. We're nonpolar. We don't have any kind of charge to create hydrogen bonds, and so we don't like to be around things with charge. So we are repelled by our water. This is your oil and water example. So oil and water just don't mix, because oil is non-polar and therefore hydrophobic and is repelled by the water. Last thing, promise, last thing I'm going to piece, because we're almost at an hour here, is that we have an ability for water to sometimes transfer hydrogen atoms between its structure and create individually hydronium ions, which have positive charges, and hydroxide ions, which have negative charges. Now, hydronium ions are usually just broken down and represented by a hydrogen so a positive hydrogen or a hydrogen ion. So hydroxide, we keep it as hydroxide, but these are also very important because they are very reactive, and these allow a lot of different things to happen. They will pop up very frequently as we progress through the next few units. So we have some specific solutes that can disrupt the balance that we have between our hydrogen ions and our hydroxide ions when we're dealing with pure water. And so these come in two varieties. We have acids, which increase the amount of hydrogen ions we have in the water, and bases, which reduce the concentration of hydrogen ions that we have in the water. Essentially, they're increasing our hydroxide ions, but we look at this specifically from the gaze of hydrogen ions, because a measure of the concentration of hydrogen ions within our solution is its pH. Now, pH is another one of those equations that they give you on the AP formula sheet, but I want to put your minds at ease that you will not be asked to determine the concentration of we won't have to determine the pH of a solution. But we do want to understand what pH is. So pH is the negative log of our hydrogen ion concentration. So it is one of those pieces that increases tenfold each time we move a step. So we have 10 times more hydrogen ions in stomach acid than we have in lemon juice. So we have less hydrogen ions as we move up into the greater numbers. We become more basic or more alkaline. This will come up a couple of different times, specifically when we are looking at the differences in concentration or the concentration gradient between different sides of a membrane. But I have ranted and raved for far too long as it is. Thank you guys so much for hanging in here. This will probably be one of our longer videos because of all that chemistry information I wanted to make sure that I added in for those who maybe needed a little bit of a refresher or those who haven't taken a chemistry class yet. With that being said, I will leave you guys here, and I hope that you guys feel comfortable with this information as we work through it this week, and I will see you in the next video.